This is episode 268 of the Stem Cell Podcast, Tissue Formation, with Dr. Shukry Habib. Hello, everybody. We are Daylon and Arun. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The show is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the podcast, please rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Shukry Habib from the University of Lausanne. He's on the podcast to talk about his research on tissue formation and repair. We've also got our usual roundup of recent highlights in stem cell news. That's coming right up.
But first, ICCR 2024 is only 6 weeks away, and the scientific content is organized around 6 tracks. Clinical applications, number 1. Number 2 is ethics, policy, and standards. Number 3, pluripotency development. Number 4, disease modeling and drug discovery. Number 5, new technologies. And number 6, somatic stem cells, cancer, and regeneration. Visit iccr2024.org to check out the program in more detail, and we'll see you in Hamburg. Of course, ICCR, the annual meeting, is all about the coolest new advances in stem cell biology. And, we talk about this all the time. One of the coolest current advances in stem cell biology are these early embryo models. We almost talk about it every single show. And last year, one of the pioneers in the field of early embryo models, Magdalena Zernicka-Goetz, gave the opening plenary, the opening keynote of the ICCR 2023 meeting, which is a fantastic talk about some of her work that they've been doing in her lab over the last, however many years to establish these cutting edge early embryo models for mammalian development. And so on that same note, the first Roundup paper from this week is going to be a paper from none other than Magdalena Zernicka-Goetz over here at Caltech in my neck of the woods. This is a very basic study, but I think one that has, and I'm sure you'll agree with this, some implications for IVF and potentially how we might be able to improve it. This is a Cell paper titled "The First 2 Blastomeres Contribute Unequally to the Human Embryo." This is a very fundamental study, a beautiful observational study that was done by the Zernicka-Goetz lab. And the concept is really simple, right? Like after fertilization, when the sperm and egg meet to make the zygote, they're trying to figure out what happens at that point, to the initial cells that divide from that initial cell division, right? So 1 becomes 2, 2 becomes 4, blah, 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 right? But apparently they found, and contrary to popular belief, perhaps, that those 2 initial blastomeres, after the first cell division, they ultimately contribute unequally to the human embryo. And there's a portion of those that one of those contributes largely to the epiblast, and the other contributes to the rest of the tissues of the developing embryo and the support tissues as well. Really kind of astounding, and something that you may not have thought of, in terms of early development about how that happens. And to really flesh out these observations, they use some beautiful imaging and live imaging of human embryos. This is, of course, very obviously a very tough study to do because it's just so hard to obtain zygotes these days. And you know more about this than I do, but most of the time for IVF, early embryos are frozen at the blastocyst stage. And so there's actually not as much of a need to freeze zygotes. So actually just obtaining zygotes from an IVF clinic was tough in itself. I was reading some of the backstory of this paper. So, there's lineage reconstruction, lineage tracing. We've done this in various different tissue types, model systems as well. But in humans, the retrospective lineage reconstruction actually predicts that these dramatic clonal imbalances that ultimately are present in the body of the different clones arising from the ancestral origins, they can be traced to that initial 2-stage embryo right after the zygote. But the question is, where and how these clonal asymmetries arise in the embryo that's not super clear. And this is where they actually did this amazing lineage tracing of human embryos, real human embryos at that post-zygote stage, labeling them and also doing a bunch of computational predictions to determine the contribution of each of those 2 cells, those 2 cell stage blastomeres, to the epiblast, the hypoblast, and trophectoderm, right? The major germ lineages, the tissue types that ultimately comprise the embryo and it supports tissues. Of course, the epiblast is the body of the embryo proper, hypoblast being the yolk sac, and the trophectoderm placenta. And what they saw through this really beautiful lineage tracing was that the majority of the epiblast, the body proper, actually originates only from one of those two blastomeres at the initial two-stage embryo. That's pretty amazing because maybe hypothetically you would think it's an even contribution or whatnot, but it's skewed in a way. They also saw that only 1 to 3 cells become internalized at the 8 to 16 cell stage transition. Um, and part of the interesting question is how are these differences specified like early on, right? If one of the blastomere is ultimately giving rise to the body or the majority of the body, does that mean those differences are specified at the stage of fertilization, like depending on where the sperm actually ends up fertilizing the oocyte? And I think that's something they hypothesized here, and I think they didn't really flesh that out per se in this paper, but I'm sure that's the next stage of the studies that they're doing. So like what I was talking about, those internalized cells in the 8 to 16 cell stage transition are actually more frequently derived from that first cell to divide at the 2-cell stage. All right. So they're taking it way back. They're looking at those initial cell division 3, 4, 5, and seeing what those contributions are. And it's not an even contribution. And ultimately, they're proposing that these cell division dynamics and an internalization bottleneck in this early human embryo, establish an asymmetry in the clonal composition of the future human body. So it's a really fundamental study driven by some amazing semi-invasive and non-invasive imaging. Of course, they did the computational analysis to actually predict where these cells in the early stage embryos are actually moving, but they also injected them with some GFP expressing mRNA to track individuals' blastomeres over the first few cell divisions. Really beautiful fundamental study. But like what we were alluding to at the beginning, I think it could have some implications for IVF and being able to identify perhaps the successful blastomeres early on. Perhaps the blastomeres to prioritize in those IVF screens, perhaps. I don't know. I'm just, spitballing here is a little bit. But I think, like I talked about, the thing that really excites me is the next step of this is being able to figure out, okay, we figured out that one of the blastomeres is having an uneven contribution to the body proper. The question is why? Is that because of where the sperm actually enters the egg? Is that driving the whole asymmetry process, even at the 2-cell stage? Really cool conceptual advance here.
Agree, man. Conceptual advance here is big for me as someone who really appreciates and focuses on pre-implantation development. I heard you pepper the word a few times around there in your description there, amazing. And you can't spell amazing without MZG. And this is just another point of evidence for me that the notorious MZG, as I sometimes like to call her, is indeed the big mama of pre and peri-implantation human development. And for me, as you said, the conceptual advance is huge because, the consensus, I guess, and understanding is that there's, vis-a-vis the 2C-like cells, is that the 2-cell stage is 2 blastomeres where there is perfect equivalency, right? And that to me just underscores a couple of things here. One, the power of observation and the value of just basic observation here that is still deployed in the midst of all these super high tech tools that we have. Just looking at a thing, observing a thing is still critical. Also, the need to work with pre-implantation human embryos still. People thought that since iPS cells, I thought that, we don't need blastocysts anymore for generate stem cells, but it's still key to understand these pre-implantation processes for advancing reproductive medicine, for fundamental understanding of pluripotency and totipotency and where that boundary lies. But more than anything, to me, it's just the courage of the notorious here and just going after something. And of course, she's poised to do it with the profile that she has. But it still, I think, has taken a tremendous amount of courage dedication to get to this point and to really have played a large part in giving birth to an entire field of study and insight. So my hat's off to the Zernica Goats Lab here for another really pioneering study that is more than anything resetting our understanding, I would argue, of pre-implantation development at the earliest point of differentiation.
Yeah, literally the first cell division. You can't get earlier than that, right? And I think I agree with you that Magdalena has really been a pioneer in this field and helped push it forward in the last few years. But a study like this, I think, is uniquely enabled by where she is, Caltech, right? Because this is a tech-driven study in large part. It's not it is observation, but it's really fine observation based on these custom microscopes that they build, the computational analysis that they actually doing. And to be completely honest with you, we're just down the road from Caltech here at Cedars-Sinai. We actually had a symposium 2 weeks ago where we wanted to figure out ways where we could harness Caltech's expertise in the tech, right? Because they're amazing at basic science and they're amazing at tech development. And as a medical center over here at Cedars-Sinai, we want to uh, synergize with that. We want to work together with them on that, using some of these cutting edge approaches and computational modeling to understand medical concerns and translational concerns as well. So it's, of course, building on decades plus of Magdalena Zernica Goetz's work. And I think she actually alluded to it in one of the news and views associated with the article. The framework for this was established in some mouse observational work that she published more than 2 decades ago in the early 2000s. And now this is 2 decades forward, focusing on the human. But like I'm talking about, this is enabled and this is happening now because of these amazing developments, computational modeling, predictive imaging, all these kind of things. So shout out to Magdalena and her lab for just another incredible foundational paper in this field.
Yes, that is a good point. We've come a long way since von Leeuwenhoek. What a callback. Observing sperm, we're now looking at things, observing, sure, but as you said, with a much higher degree of resolution. So yes, again, kudos to Magdalena and her lab. Going back to sperm, I got a story that even precedes the two cell stage, if you can believe that. We're talking about gametes here, and actually talking about gametes from pluripotent stem cells, which has long been a long sought holy grail, I guess you could say, for reproductive medicine. So we've talked a lot about this in the show. We've talked about the players who have driven it, namely Hayashi and Saito. It's a paper from Saito Lab here that was published in Nature, and we're talking about human primordial germ cells. So just a bit of background, specified around embryonic day 12 to 16 in human embryos in the amnion or posterior epiblast. And then over the course of around 4 to 5 weeks, they migrate through the yolk sac and the hindgut endoderm, and they ultimately colonize the genital ridges where the gonad actually will form. And during that period, those human primordial germ cells, they undergo a lot of reprogramming, right? This epigenetic reprogramming that resets the parental epigenetic memory, as they call it, through this genome-wide DNA demethylation and histone remodeling. And then by around 7 to 8 weeks post-fertilization, those primordial germ cells have completed the reprogramming and they differentiate either into the prospermatogonia in males or the oogonia in females, which are precursors, of course, for sperm and oocyte differentiation. So in terms of what we can do with pluripotent stem cells, there's been a tremendous amount of progress in the last decade or so, and culminating in the generation of human primordial germ cell-like cells from pluripotent stem cells that when you complex them with embryonic testicular or ovarian somatic cells, in this case, they do this xenogeneic reconstitution with mouse, what has been done so far with mouse embryonic testicular or ovarian somatic cells, they can actually get the primordial germ cell-like cells to undergo that epigenetic reprogramming and differentiate to those spermatogonia or oogonia-like cells, respectively. But the approaches are very low efficiency and not scalable, of course, because you got to get that somatic tissue every time, or even translatable, you might argue, given that you got to find a source perhaps of human fetal uh, gonadal somatic cells, or complex it with mouse or another species, which I don't know if that's going to get by the FDA, considering that we're making germline cells here. I've talked about that in the past. Um, so if we're gonna, make this process translatable, you really gotta have a more robust methodology, and that's where this story comes in, which is really a bombshell. This is at the point that I'm reading, it's a accelerated article preview, and it merits that because a lot of people are really reading this with tremendous interest. In this story, Site2 Lab established a strategy for recapitulating that epigenetic reprogramming and differentiation of those primordial germ cell-like cells into mitotic prospermatogonia or oogonia. And the key here is that they coupled that with extensive amplification. We're talking more than 1 billion fold, so truly unlimited amount of putative germ cells here. And what they found was surprisingly, as they say, well, I'd say surprisingly, given our guest and the running joke on the show, it wasn't centered on Wnt signaling. But jokes aside, it was driven primarily, not primarily, but a major player here was BMP signaling, a key driver of the process. And mechanistically drilling down, they found that it involved attenuation of MAP-ERK signaling, as well as DNA methyltransferase activities and resultant DNA demethylation. And what they found here, and this is so cool to me, is they showed that if you had deficiency in this TENS11 translocation, or TET1, which is a DNA demethylase that's abundantly expressed in human germ cells, if you get rid of that, these PGLCs, they differentiate to extra embryonic cells, including amnion. And that is coupled with de-repression of these, bivalent poised promoters that drive that fate. And at the same time, in those cells, they fail to activate the genes that are vital for spermatogenesis and oogenesis. And in those cases, those poised promoters remain methylated and repressed. So for me, I clearly have a bias, but what is so amazing about this story is that this has been like primarily, I guess, an experimental result that has been derived from the xenogeneic reconstitution experiments. Here, it's taken to a level of mechanistic understanding where you really could conceivably drive germ cell differentiation from human pluripotent stem cells without having to do these complicated tissue reconstitution approaches. You can drive it really just by gene expression and manipulation, or perhaps having a defined cell type that could provide that paracrine influence, whatever it is. It's a major milestone for human in vitro gametogenesis research. And, I would say perhaps has potential to be translated. Maybe you could have in vitro gametogenesis from pluripotent stem cells that'll generate babies. But no shade on this story. I love this field, and I love the potential and the potential for scientific understanding, medical application, etc. I am very wary of the idea of taking any cultured cell and incorporating it into the germline of our species. I've talked about it a lot, but nevertheless, this is a bombshell study that is going to be lighting up the servers.
So I was actually about to allude to that because I know it's a touchy subject for you in terms of making these things and actually using them for reproductive purposes. I mean, I agree with you first of all that this is a bombshell. It's you're talking about that epigenetic reset. This is a big limitation that they're able to overcome here. The expansion part of it is great. The just however many fold they're able to grow these things. Uh, Eliminating the xenogenic part of it, I think has a lot of potential as well, avoiding some of those co-culture applications or co-culture requirements that some of these studies had to have in the past. Because like you're saying, if you do want to bring it to a translational reality, you can't have that xenogenic co-culture. That's just not that's just not the FDA is for one. It's never going to approve that. So having an all human, all single species culture is great. So with all that being said, that framework, does this change your perceptions of the next step in this field? Do you think this is overcoming some of these scientific barriers? Does it make you more amenable to making this a reality? Are you more okay with it now?
If I have to be completely honest, it's scary to me. Scary because I guess it was a comfort to me that it didn't seem practical with the xenogenetic. That was an easy flag to send up, red flag, I'd say, to say, maybe we should be more cautious on this. But now what gives me pause is that number. Over a billion-fold amplification. Wow. That's a lot of expansion. That's a lot of clonality of indeterminate potential. So I have major, I guess, I don't want to call it concerns. I don't think I'm qualified to speak to whether or not this is risky. I think my intuition tells me that we got to do a lot of long-term, primate level studies to see how this plays out in terms of like completely in vitro reconstitution. That said, alluding back to what we were just talking about in terms of like the amazing resolution of the tech nowadays, I guess like an early endpoint that you could look at, you look at whole genome sequencing, you do it in enough embryos, and perhaps you can arrive at a degree of, I don't want to say certainty, but comfort that you're not introducing a lot of, I don't know, indels, but aberrations in these downstream gametes. But I don't know, Arun. I think it's really tremendously exciting. I also know that there's a lot of IVF patients that would really do anything to have a baby. And I wonder about our species.
Wow, how do I follow that? Yeah, hey, I mean, whether regardless of the clinical translational applications potentially associated with in vitro immunogenesis, I think the thing that excites me is the feasibility, right? This is becoming more of a scientific reality. And that in itself is super cool because the mouse field is so far ahead when it comes to IVG, but human is certainly catching up. So again, just from the basic science perspective, I think there's a lot to be excited about here. So moving on to something else, obviously that's very exciting in the stem cell field, and that's organoids. If we're talking about two things here on the show, it's either embryo models or organoids. That's the running joke, right? In addition to Wnt signaling, I suppose, which unites everything. This is a Cell stem cell paper titled Modeling Blood-Brain Barrier Formation and Cerebral Cavernous Malformations in Human PSC-Derived Organoids. This is coming from Ziyuan Guo, apologies, from Cincinnati Children's. And as we know, Cincinnati Children's, in my mind, is perhaps the world's leading world's leading institute when it comes to organoid biology. I mean, the things that they've done over the last decade plus in organoid biology are astounding. I mean, you pick a tissue type, you name it, they've made an organoid out of it and have used it for drug screening application, disease modeling applications, whatever. I think honestly, it is the place to go for organoid biology. Shameless plug, I suppose. I have no affiliation with Cincinnati Children's, but I love the work that you guys do. So yeah, again, coming from Ziyuan Guo's lab at Cincinnati Children's, first author here is Landau. The concept here is really cool. They created an assembloid, a la Sergio Casca and his cortical assembloids, but they created a cerebral organoid in combination with blood vessel organoids and combined them to create a blood-brain barrier assembloid that could model the blood-brain barrier and permeability of the blood-brain barrier. Okay, why is this important? Well, as we know, the human blood-brain barrier is this really specialized tissue that regulates passage across the blood and central nervous system compartments. And I'll get to one of the caveats from this study in a little bit. I'll go ahead and mention it here. One thing the blood-brain barrier is really important for is drug permeability, right? There's certain drugs in the circulatory system that cannot cross into the brain because of this blood-brain barrier. That's actually one thing, surprisingly, they didn't dive too much into in this study, because when I think of blood-brain barrier, that's the first thing I think of is drug permeability, but we'll talk about it. So, we know about this critical physiological role of the human blood-brain barrier, right? But the models are okay. And I don't want to get in trouble here because my boss here at Cedars-Sinai actually made one of these blood-brain barrier models from an organ chip. They actually published it in Cell Stem Cell a few years ago. Clive Svensson, same senses, just down the hallway from me here at Cedars-Sinai. They had this, 3D blood-brain barrier on a chip where they were able to actually model drug permeability by having neurons on one side of the chip and brain microvascular endothelial cells on the other side of the chip. But these folks, they're trying to build on that, right? So they're saying that they're making an organoid model of a blood-brain barrier, something that can perhaps better mimic blood-brain barrier development, which is something that the chip studies didn't do, as well as the human blood-brain barrier function. So here they created these human blood-brain barrier assembloids from brain, cortical, and blood vessel organoids, just kind of smashed together. Ultimately, that were derived from pluripotent stem cells, and they validated the acquisition of these, blood-brain barrier specific signatures, whether it's the permeability, the molecular, transcriptomic. They did a ton of single cell, as you would expect, to actually characterize these organoids, the cellular functional characteristics as well, and also uncover this. Importantly, a neurovascular crosstalk. So there's these two types of organoids that are actually talking to each other: the blood vessel organoids, the cortical organoids. There's a crosstalk and a spatial patterning within these blood-brain barrier assembloids. And then I think the reason that this ultimately became a stem cell paper, even without the drug part of it, they did a patient-specific disease model. Okay, so they derived patient-derived human blood-brain barrier assembloids to model cerebral cavernous malformations, which are a specific type of malformation of the blood-brain barrier that's arising developmentally in some patients. And they actually found that these assembloids were able to recapitulate some of the anatomy, some of the structural defects that those patients exhibit with their, the blood-brain barrier found in their brains. As well as the breakdown that happens in these patients in their blood-brain barrier. So they compared these patients-derived assembloids with control assembloids, and were able to show that indeed there's some transcriptomic overlap, with the patient assembloids with the controls, but also some significant differences in terms of the molecular signatures. Um, uh, but also, I think the cool part of this is some of the structural differences they were able to recapitulate at the morphologic level in these assembloids, replicated some of what you would able to find clinically as well. So altogether, I think a nice disease model study. This is a cell stem cell paper that's focused on the disease model. But again, I'll say, and again, not to shoot down the study at all, but I was surprised not to find any sort of drug permeability assay or experiment associated with this study, because that's a big part of the blood-brain barriers being able to block or modulate drug permeability from the circulatory system to the brain. And funny enough, when I read through the paper and noted that as the number one limitation, they actually specifically mentioned that in the first sentence of their limitation sections. We're like, yeah, we know you're probably looking for the drug permeability part of it. It's not here, but we're working on it. So shout out to this group. I think it's a really great in vitro model of this particular disease.
Yeah, this is a cool study. I love, organoids. I love assembloids. I love it all. I'm trained up as a vascular biologist in large part, so this has everything for me. I mean, neural is not my favorite, if I'm honest, but the vascular rescues it. But the combination here, I'm so pleased to see that we're talking about combining a vascular anything with a, tissue specific anything. And that's what I've been saying is a major limitation of all this macro scale 3D biology, is that it really lacks the fundamental component of scale in biology, which is vascular perfusion, right? So I was really impressed that that they were able to get that endpoint to make this assembloid with the vascular component. Although I would say, I mean, you said it, they smash them together. It's certainly not physiological in terms of how any tissue becomes vascularized. You don't grow the vascular and then the other and then smash them together. And I think that is a bit of a limitation in terms of, really recapitulating the whole of any system where you're trying to make it vascularized. But here, with the specific intent that they're going after here, with the anatomy of these cerebral cavernous malformations, I think that it's the perfect match. So it is a good fit for their endpoint. I was intrigued, as you said. The first thing they called out was the inability in this study to model drug delivery, tox, and all that. But then they followed that by offering maybe a future direction here, which is to engraft these assembloids into mice in vivo. And I think that in that context, that there would be further growth, remodeling, integration of the tissues, as well as anastomosis with the host circulation and potentially blood flow through the graft that would model like a bona fide assembloid with the vascular component that is actually functioning in a vascular capacity. So I think that that's the next step. I'm sure those assembloids are already in vivo right now, and they're just watching them. So looking forward to see what comes of that, and hoping that this serves as inspiration to other groups to make these assembloids that have a vascular component. Very, very exciting work for me.
Really cool. I totally agree with you. I think, the integration of these assembloids into the in vivo system is the inevitable next step, and it's probably already happening in the lab, because right now these assembloids with the blood vessels integrated, they're not true blood vessels, right? They don't have an active circulatory system. I don't even know if they have like a true lumen in there to carry drugs to and from the neural tissue side of it. So I think to mature the vasculature side of it, I think you would have to integrate it with the in vivo blood vessel network. The other part of this I'm excited about, and this almost brings me back to my own work, is if we can make these vascular organoids, then there's just an infinite number of tissues to which you can apply them to, right? I mean, vascularization of organoids is a problem across the board, no matter what or type of organoid you're trying to derive. But if you're able to bring in the vasculature from a vascular organoid and combine it with, say, like a cardiac organoid or like a pancreatic organoid, whatever, that's the goal, and that's ultimately the dream, to make these things more complex, make them larger, give them an integrated vasculature, circulatory system, whatever you want to call it. And then, yeah, if you want to take it even one step beyond that and make them even more mature, you have to integrate that assembloid in vivo. I think it establishes like a nice workflow almost for using vascular organoids specifically for whatever cell type or tissue type you're interested in.
Yes, I mean I guess that's the unmet need in the organoid field, is a vascular component. But I don't know. Could be forever. Could be in a month that we're addressing that. I think the jury's still out, but um, I think we're getting there, and this is evidence to that point. And that's our goal, right? We're talking about taking human-induced pluribone stem cells. That was the kind of the root goal, ground truth there, was that we're going to make these cells into tissues. But a lot of people in the adult stem cell field are probably rolling their eyes, like, why are you trying to reinvent the wheel? There's a lot of stem cells in the body right now. Why don't you just deploy those? And I'm going to tell you in our last story about that logic and that approach. And this is kind of a sequel to a story, same journal, Cell Stem Cell, same lab, which is Li Jianhui at the Chinese Academy of Sciences. A lot of the same authors, um, and the same fundamental approach. The first story, it was episode 265 we covered with Ryan Flynn. It was about liver failure. This is about liver disease, genetic liver disease, and it uses a lot of the same approaches. But just to review, we're talking about using cells for treatments, right? To repair damaged tissues, and the liver is a huge unmet need there. But the efficacy of these allogeneic hepatocyte transplants it's not great, right? Because you either liver transplant or actual hepatocyte transplant that are encapsulated, you have to deal with the immune rejection, right? That's the fundamental obstacle there. Even with immunosuppression, the engraftment is not really efficient. So the idea of using autologous cells, obviously an advantage there, particularly when you're talking about curing genetic disorders, right? This is not really even liver failure. There's no fundamental limitation in terms of the niche. It's really just hepatocytes that aren't really working well, and they need to be corrected genetically. As I alluded to, there was this recent story from Legion's lab using these autologous hepatocyte cells that were primary hepatocytes that were ex vivo cultured for really extended periods, proliferating. They de-differentiate them and they expand them. Um, and they use the same approach here. But the key here to this story, which I think merits its coverage and merited a second publication in Stem Cell with largely the same technology, is that even so, doing knock-in, doing any kind of genetic correction in primary hepatocytes it's really inefficient with the current state of the art and CRISPR-Cas, whatever you're using. And part of that has to do with the low proliferation capacity of primary hepatocytes, which this group kind of solved, but also has to do with the adeno-associated viral strains that are used for CRISPR-Cas. It's just not one that's been optimized. And this story, very straightforward. They use the same tech, which, be my guest, go back to episode 265 and review. I did a more lengthy description of that approach, but it's pretty basic. As I said, they take out primary hepatocytes, de-differentiate them, expand, and then differentiate them back, show they have function here. They do the same approach, but then they've optimized after systematic testing and optimization of numerous adeno-associated viral variants. They identified this AAV 2.7M8 variant that enabled the CRISPR-Cas9 mediated targeted integration with high efficiency. And they were able to correct the pathogenic mutations in FAH, which is fumaryl acetoacetate hydrolase, as well as OTC, ornithine transcarbamylase. Those are two enzymes that are implicated or have been identified to be the basis of genetic liver disease. And they, with really amazing efficiency, Arun. I invite all you guys to look at these pictures because they were able to repopulate after injury to clear, able to repopulate the mouse liver. You look at these pictures, I would argue it's like more than 50% of these mouse livers are composed of human cells. And those mouse liver, proles, they call them. I'm not going to go through the acronym, but they matured and ultimately treated and corrected mice with tyrosinemia. So, this is one of the, I guess, less appreciated successes of recent years because we're so focused on these really difficult endpoints of maturing induced pluripotent stem cells while differentiating them and getting them integrated in tissue, all really tough challenges here. And studies like this, I think, are showing the power and translational potential like tomorrow of adult stem cells, in this case, the liver. So, given the tremendous unmet need here, I think that this, along with like those xenotransplants, which I know the kidney patient just died, and I wouldn't call it a setback, but I guess maybe dampened enthusiasm in that field. I still think that and this and studies like this deploying adult stem cells are really the immediate future of regenerative medicine and are going to change the world as we know it.
Yeah, it's, I guess, a low-hanging fruit, even though the work is still extremely complex that they have to do here to make this translational dream a reality, even with the adult side of things. But it's not perfect. Certainly, anytime you're talking about genome editing, you have to make sure that the cells you're editing they don't have any off targets, right? It's something we've talked about forever, especially in the Cas9 field, and we know. As the analogy that I always make here on the show is taking a shotgun to the genome with Cas9. And there's, of course, newer generations of these editors to alleviate some of those off-target problems. But I mean, they directly mention it in the study. So, this there were chromosomal translocations like they actually found in the ABS1 editing site in the study, which is obviously something you don't want. Maybe because of the spatial proximity between the two sites that they're trying to edit. And they say it in the future, they want to have a safer AAVS1's guide RNA site with minimal off target. Whether they're able to identify that bioinformatically, and there's a billion tools that you can use to actually identify safer guide RNAs, or maybe use a different type of editor. I don't know. I think there's a lot of future approaches that they can take to make this approach even safer and more effective, at least when it comes to the genome editing side of it. And the other thing is, of course, and this is minor, they use actually GFP as a selection protein for cells of successful genome editing, but of course you can't really use that for FDA approved applications and clinical translation. So plenty to work on here, but I think a nice proof of concept study in this field.
Yeah, that's a good point. I mean, I should just chill out a little bit with my tomorrow claim there because, yeah, we're not just dealing with the indels of when you're trying to knock something out, but in order to get a knock in of a gene, I think that it's a higher bar, and you really have to make sure that you're not introducing some pathology there with the cure. So recognized, Arun. Thank you for mentioning that. And I think, as you said, there's going to be a lot of tuning in the same vein as they did. I mean, they really went in. Half of this paper, more than half of this paper, like figures 1 through 5, are all about the adeno and how they came to it. So I think that they're familiar with optimization, and there's groups all over the place that are coming out with new tech every week that's optimizing these gene targeting approaches. So I think it's a tractable problem. And also, just to expand the scope of this a little bit, we talk about historically, people love to use the liver as like a little biorepository. Hemophilia, the idea was transducing liver with factor VIII to try and rescue hemophiliacs. Like, if you can get these highly proliferative hepatocytes, that's like a biorepository, I think, that you can introduce in vivo as a therapeutic. So, notwithstanding, of course, or recognizing, I should say, the risk of any kind of gene knock-in, I think that maybe this fundamental approach of finding a good adeno that can target, as well as finding cells that are targetable and highly proliferative, is a really useful combo in terms of I don't know, maybe like a biotherapeutics with a liver as the producer there. I don't know. Could be science fiction, but what isn't science fiction that we talk about on this show? I don't know. That's the end of the roundup. Moving on to the interview in just a few seconds, but before we get there, a quick message from STEMCELL Technologies. As research using pluripotent stem cells advances toward the clinic, there is a renewed focus on cell quality. Visit www.stemcell.com/cell quality to explore ways to assess your human pluripotent stem cells and learn about essential quality attributes to consider for gene editing, disease modeling, and maintenance. All things apropos to the Roundup stories we talked about today and our guest, who's coming right up. All right, everybody. With us from Lausanne University, where he is associate professor in the Department of Biomedical Sciences, we have Dr. Shukry Habib. The Habib Lab works at the intersection of stem cell biology, biophysics, and tissue engineering to investigate how tissues form. They use this knowledge to develop technologies for tissue repair. Shukry, thank you so much for joining us on the show today.
Thank you very much for having me.
Yeah, it's great to have you here. I mean, like Dylan mentioned, your lab really focuses on the internal and external cues that regulate mammalian stem cell division, bait choice, all these very fundamental mechanisms, right? It's a really broad topic. I think has a lot of different applications, implications for engineering too, which is something that you've delved into, engineering better organized human tissues for in vitro modeling, maybe even for transplantation. So to start things off, could you just give us a broad overview of what your lab is working on right now, and both on the basic science and also the engineering side of things?
Yes, sure. So my lab works indeed. We have two arms in the lab: the basic research, where we study mechanisms of recruiting, how stem cells recruit niche signals with a focus on Wnt signaling. and then the downstream effects of asymmetric cell division. And the second arm is focused mainly on tissue engineering, where we ask the question, can we use the knowledge from localized presentation of Wnt and induction of asymmetric cell division to improve tissue engineering? And here we focus on engineered tissues related to the bone. And in the tissue engineering, we also focus on developing technologies that can promote bone repair, particularly for large bone fractures or defects.
So yeah, I mean, this is a little bit outside perhaps your wheelhouse, but we've been talking so much lately about self-organization, just generally in terms of at the cell level in tissues, but also more fundamentally in embryos, like primary organization. Most recently, on the last episode that we had here featuring Shankar Srinivas, who's from Oxford. And this is timely. I mean, it makes sense because there's these unprecedented opportunity to interrogate early morphogenetic events using these embryo models, mouse, more recently human embryo models. And as I said, I know your work is focused largely on basic mechanisms of Wnt signaling in embryonic and adult stem cells, and as you said, generating these translational tools for tissue repair. But as someone with a research focus that lies really at that nexus of cell fate specification, tissue organization, and engineering, what, in your opinion, are the research questions that are going to be the most exciting ones to ask using these embryo models, either in your lab or elsewhere? What do you think is the most exciting opportunities that have been presented by these new tools?
Well, this is a fascinating field, and many people currently work on it and they try to push the limit of developing the embryo models. And clearly, one of the major question is how these embryos form, how do they self-organize, how do cells communicate and get the specification. And as I mentioned, many groups in the world work on this, and we have seen in the recent years a lot of advancement in this field.
Yeah, it's a really exciting area of study in the stem cell field. I mean, we've been talking a lot about these embryo models here on the show, and I'm sure they're going to be covered extensively at ISSCR. But focusing a little bit more about the work that you're actually doing in your lab as well, you had this really cool Nature Materials paper that I wanted to talk about a few years ago, where you developed this transplantable bandage that actually secretes Wnt for inducing bone repair in vivo. I think it's a really exciting system. It represents a nice snapshot of what's possible in the stem cell field right now, where you can appropriately intersect bioengineered approaches with these fundamental basic mechanisms in molecular studying, molecular signaling as well. So tell us a little bit more about this work, this translationally engineering oriented work, and maybe where it's on the road to clinical translation, if it is at all. And I know you've got a couple of other things in the pipeline as well, so feel free to talk about it.
Yeah, thank you very much. So indeed, this work that we published a couple of years ago was led by very talented group members, Yoshi Okochi in my lab, and a brilliant student, Josh Reeve, who actually built this system. And the way it started, we asked the question again, how can we use our knowledge from Wnt-mediated asymmetric cell division to improve tissue engineering? And here we started the process of taking different biomaterials, and one of them is a PCL, which is biomaterial that is being often used in orthopedics, and we immobilized Wnt proteins on it to present it in asymmetric fashion to the stem cell. And here I would like to say that in vivo, the Wnt is often secreted locally and presented to one side of the responsive cell. So that was our engineering approach to make sure that we present the signal to the stem cell in a directional way. And then we took from human donors skeletal stem cells, and we put them on the Wnt platform or the Wnt bandage, and we engineered a 3D scaffold mainly composed of collagen that mimics the extracellular matrix in the bone. And what we noticed that indeed the localized Wnt would induce asymmetric cell division of the stem cell, maintain the stem cell, and generate a daughter cell that enters into the gel and start the differentiation program. And within 1 week, if we leave them in a media that promote osteogenesis, it's the process of bone formation, we could see a cascade, a very nice cascade of differentiation while the stem cells are maintained. And that was the initial story where we discussed in detail the mechanism of the formation of this 3D organoid. And then we wanted to ask, can we transplant this organoid in vivo, and would it promote bone repair? And please remember that this organoid is of a human origin. So we took the whole structure with the bandage. It's made on the bandage, and we transplanted a critical size defect of a bone. And when I say critical size defect, they have a very limited ability to heal on their own. And to our surprise, the bone within 8 weeks start repairing. And not only that, we could see bone repair, but the bone that formed was of a human origin as well as mouse, because we used only tiny amount of cells that we transplanted. So that was the 3D organoid system that we developed. And we also have the Wnt bandage alone that can stimulate the endogenous stem cells and osteoprogenitors to generate the bone. So regarding your question about taking it to the clinic, indeed, we are very enthusiastic about this work. And in the coming weeks, I will be having discussions with different orthopedic surgeons to start discussing how we can bring this bandage to the clinic. And clearly, we would like to talk with the regulators here in Switzerland to see what is needed for us before we proceed to the next step.
So yeah, I mean, I get that ultimately we're all looking toward the translational application. I mean, collectively speaking, I think a lot of our work, specifically in the stem cell field, that's kind of the ground truth, right? We're doing this for translational purposes for the most part. But we also know that the best science culminates in unraveling basic mechanisms, right, that underlie cell biology, embryo development, tissue morphogenesis, and you never know how those insights may feed into the clinic. You've recently been focused on these cytonemes, right? These thin cellular projections that mediate exchange of signaling proteins between cells and their role specifically in modulating signaling activity between the trophectoderm and the epiblast cells in early embryos. Would you elaborate on that a bit and underscore how these basic signaling mechanisms are important for understanding and controlling differentiation of pluripotent stem cells, perhaps toward a translational end?
Yeah, sure. So indeed, when I established my lab at King's College London after the postdoc, one of the question was, how do the stem cells find their niche, and how the niche communicate with the stem cells and vice versa. And to simplify our approach, we used a bioengineering approach where we know that embryonic stem cells rely on Wnt proteins for self-renewal. So we immobilized the Wnt on microbeads, and we put it in a closer proximity to the stem cell, but it wasn't touching. And what we noticed that the embryonic stem cells will extend these cytonemes that you mentioned, which are thin filopodia, signaling filopodia, and try to sense the environment. And once it finds the localized Wnt that is required for the self-renewal, because we put different type of Wnt's, it is equipped with receptors to recognize the localized Wnt and then recruit it to start the process of asymmetric cell division. This all connected when the work on embryo models start appearing, where they showed that it was a work from Magdalena Gutz's lab, they showed that if you take a trophoblast and embryonic stem cells and put them together, they have the capability to self-organize, self-sort, and start forming early embryonic models. So we looked if this same process that we observed between the embryonic stem cell and the bead would happen as well between the embryonic stem cell and the trophoblast stem cell. And indeed, we found that the trophoblast secretes locally many Wnt ligands, and the embryonic stem cell will move around and send these cytonemes and start sensing the environment in close proximity to the trophoblast. And once they recognize that there is Wnt, they will send a large cytoneme and start the pairing process with the trophoblast to start forming the 3D embryonic model. So for us, that was a fascinating observation, and we even found that there is a lot of similarities between the interaction of the localized Wnt or the trophoblast that secretes the Wnt and the embryonic stem cell with the neuronal synapse, because we found that glutamate receptors and calcium transient are also involved in the process of the recruitment of the Wnt and the pairing process. I think that this, to answer your second question, how I see it for translation, this is a basic mechanism of how tissues can form and how the stem cells interact with the niche, because many mammalian stem cells rely on Wnt beta-catenin pathway for self-renewal and regulating their function. And we found by looking at the RNA-seq database that many of the mammalian stem cells in different organs also express glutamate receptors, which could indicate that similar mechanism might occur as well in adult mammalian stem cell. So you can imagine that we can use this knowledge, for example, to enhance the interaction between the stem cell and secreted factors, or with the niche for tissue engineering to improve the formation of organoids and engineered tissues.
Yeah, it's I think a lot of really exciting applications there. I think one of the morals of the story here is that Wnt is everywhere. Wnt is ubiquitous, right? It's really one of my favorite signaling pathways as a developmental cardiac biologist. We have a running joke here on the show that everything in biology seems to come down to Wnt signaling. But given your track record in this field, you might actually agree with that statement. And side note, a few of your recent Wnt papers were led by Molly Lowndes, who actually went to grad school with at Stanford, is actually now a professor of her own. And before we actually, get a little bit more application or engineering oriented, or talk a little bit more on the translational side of things, could you actually just reflect on the power of Wnt signaling in general as just one of those critical signaling axes in biology?
Yes, Wnt signaling, as you said, it's everywhere. And it's involved in early development, from determining and breaking cell asymmetry to determine later the body axis and patterning the tissues. It's important for regulating the stem cell function from self-renewal, and also involved in differentiation. And as we all know, in many types of cancer, Wnt signaling, or components of the Wnt signaling, are mutated. And on top of that, what allowed us to culture stem cells in vitro and start working with organoids and engineered tissue was because of the capability to purify Wnt proteins and add them to the media that allowed the researchers to expand the stem cells, characterize them, and use them later for organoids or other applications. So there has been a lot done in this field, and there is still a lot of things to be done here to ensure that we have a better use of the stem cells for therapies and for modeling diseases.
Yes. I mean, Wnt, it all comes down to Wnt. It's going to be the the end game is wind's gonna be involved, is my guess. All right, so Shukry, tell us, I mean, what was, would you say, the critical breakthrough for you personally? You know, the watershed for you in your career that really started you along this path and drove your fascination and interest and productivity in unraveling the mysteries of wind signaling?
So when I came to Rule Nusayed's lab at Stanford for interview, during the interview, Rule mentioned to me that they were able to purify different stem cells from different organs and to culture them in vivo. And their secret uh, recipe was to put purified Wnt in the media, and Wnt will induce Wnt beta-catenin pathway in the stem cells, which was apparently important for the expansion of the stem cells and the self-renewal. And during the interview, I mentioned to Rule that I read different papers in Drosophila embryo as well as in C. elegans, where Wnt proteins, when they visualize them, they seem to be secreted locally and presented in asymmetric manner to the responsive cell. So I asked Rule during the interview, what could be the effect of this asymmetric presentation of the Wnt to the stem cells, in contrast to what is currently being done, which is added globally into the media. And I remember Rule was silent for a couple of minutes, and then he told me, why don't you come to my lab to do this as a postdoc? And indeed, here my journey with wind signaling started. It took me 1.5 years, 2 years to immobilize the hydrophobic protein. Wnt is known to be insoluble, not very easy to work with, but eventually I managed to immobilize it covalently to tiny microbeads. And after I confirmed that it's biologically active, I chose a stem cell which is naturally not polarized, the embryonic stem cell. And once I put the Wnt, the localized Wnt, close to the embryonic stem cell, a fascinating thing happened. I could see that by using different markers and live imaging and reporters, that the localized Wnt would polarize the embryonic stem cell at the level of the Wnt beta-catenin pathway, as well as the PAR complex. And then in collaboration with Eric Bitzig at Genelia Farm, we could see in 3D at high spatial temporal resolution that the localized Wnt not only polarize the cell, but also orient the spindle. And by using different reporters, we could see that before the completion of cytokinesis, there was compartmentalization of the stem cell markers, the activity of the Wnt beta-catenin pathway close to the side of the cell that close to the Wnt, and the distal part had lower activity, lower stem cell markers. And eventually, after the cytokinesis occur, we had 2 daughter cells and asymmetric cell division. One retains the embryonic stem cell fate close to the Wnt bead, whereas the distal one start the program of differentiation, and it is farther from the Wnt bead. So this phenomena and the presentation of Wnt in a way that mimic what we see in vivo led us to discover how localized Wnt affect the cell fate of mammalian stem cells by inducing asymmetric cell division. And later in my lab in England at King's College London, we use this to also not only induce asymmetric cell division in human skeletal stem cells, but also to engineer the 3D organoids in a very controllable way and very organized within short period of time, 1 week, that can be transplanted in vivo and generate humanized bone.
Arun alluded to your time at Stanford. I mean, you've had an amazing tour of the greatest institutes, you know, from Technion, Stanford, HHMI. Your research training pedigree, truly outstanding. Your postdoctoral work in particular, which was performed with guidance from Noel Roos, who's a patriarch of Wnt signaling back when Wnt wasn't involved with everything. He was still working on it. He pioneered there. And one of the early winners of the Breakout Prize also, I should note. Also, You work with Nobel laureate Eric Betzig. Of course, every single training experience, I should say, is unique, right? And the success or failure, I don't know what you want to call a failure or a success for that matter, but it derives from a complex interplay of things, right? The individual aptitude and experience of the investigator, the personality and the group dynamic, even support from the laboratory, support from the institute, also funding agencies. This is a million things, as well as all these other inscrutable factors like courage, I guess you'd call it, grit, luck. But I'd be willing to venture that the success and prominence of the primary mentors is highly predictive of a quote unquote successful research career. I don't know. I think we're all successes in that we're doing science for a living. It's an amazing privilege and opportunity. But I just want to ask you, as someone who's operated under these real icons, what did it mean for you and your research direction to train with these super elite scientists for whom, like, there was no question that was beyond their grasp and ambition? What was that like for a young person? Did it, do you think, really shape the way you ask questions, going more, I guess, aggressively? I don't know. You tell me. What did that do for you and your career and your experience?
So I was really fortunate that throughout my career I worked in different places in the world, in different labs, and interacted with many different scientists. And I owe my success, as you said, to many of my mentors, starting with my master degree mentor, with whom I am starting now to collaborate with, Professor Abed Azim at the University of Tel Aviv. Who until now we maintain a good relationship, and he has always been there for me whenever I had a question, any challenges. He was always there to support me. The PhD was indeed under a major icon, or the god of mitochondria in Munich, Walter Neupert. I couldn't explain to you or uh, find the word to describe how happy I was to be accepted to his lab to do my PhD, where I worked on mitochondria biogenesis. And after I finished the PhD in his lab, I discussed with Walter Neupert what is, in his opinion, the next field that I should work on it. And I had different ideas, but Professor Neupert was very clear with his answer. He said, stem cell, regenerative medicine, that is the next unfolding field, which is really exciting. And clearly, I followed his advice and I applied to different labs. And eventually, I found myself working in the lab of Roel Nusse at Stanford University. It's a great honor that I was trained in that lab. He taught me the importance of being very focused. During the postdoc in the earlier years, I wanted to do many stuff, but he took me aside and he said, it's really crucial that you be focused on one specific question and try all your best to answer it. So that was a great advice from him. And Roel Nusse is well known for his amazing presentation, and I owe also my capability to present well my research in talks, seminars, and teaching to Roel. He inspired me there. And also, it was Roel when I went with him to Genelia Farm for a meeting, and Eric Betzig presented his microscope, the Bessel beam plane illumination microscope. And I remember Roel telling me that we need to approach this guy and work with him. And indeed, we immediately formed a collaboration with him, and I start working in Roel lab and then go for several months to Eric Betzig to visualize in a 3D using high spatial temporal resolution, the wind-mediated asymmetric cell division. And what I can tell you about Eric was always interested in all what we have been doing in his lab. He's hands-on, and he loved discussing science. So again, it was a great pleasure for me and a great honor to work under all these amazing scientists who helped me to pave my way in science and become the scientist who I am today.
Hmm.
Yeah, that's a great story, a great recap of your journey. And like, I totally agree with you. We're all here and doing what we're doing because folks believed in us and gave us opportunities to shine. And you're no stranger to, like you're alluding to, jumping across the world to pursue training in the best places. And now you're at the Department of Biomedical Sciences at the University of Lausanne in Switzerland, an amazing institution in Switzerland, and a growing powerhouse in biomedical research. I mean, a couple of fun facts that I found about the school. One, the Swiss Institute of Bioinformatics is apparently located right up there on the main campus. And two, and I thought this was really fun, it's also home to Dr. Jacques Dubochet, who received the 2017 Nobel Prize in Chemistry for developing cryo-electron microscopy, a pretty amazing technology in itself. And he apparently worked there for 20 years and is now an honorary professor there. And also, and this is this is really cool, he has a special Nobel laureate only parking spot for his bike on campus. I saw a picture of that. So tell us about the University of Lausanne and what's it like to work there with these amazing people and what makes it such a special place for biomedical research.
Um, uh, First of all, you need to come here to see it by yourself. The university or Lausanne itself is located in a beautiful scenic place. The university has one part close to the Lake of Geneva, and we have amazing sport facilities there. For students, for faculty, and part of the biology department is also located close to the lake. I am in the campus, what we call it, the SCHUV campus. It's the hospital. It's a bit farther from the lake. It's in the middle of the city, in the downtown, and Lausanne is small in comparison to London, clearly. What makes this institute really unique? When I was recruited to the University of Lausanne, I remember many people reaching out to discuss with me about the potential recruitment and why should I come. And from medical doctors to scientists, it was really amazing and very, very welcoming. After I gave my talk and I got selected, it opened the door to me to communicate with all these great scientists. And the University of Lausanne is very international, and it has a lot of collaboration also with APFL, which is also another great university here in Lausanne. So you can say Lausanne is a university town. The university has a lot of amazing facilities, and as I mentioned earlier, we are fortunate that part of our staff salaries and consumables are core funded. All this makes life and doing research easy or easier, not easy, easier. And the collaboration within Lausanne, within Switzerland, is very convenient. And it's a great place also to do international science and collaborate internationally.
Yeah, I mean, a lot of takeaways from this discussion. One, which I already knew, went rules, but also I really enjoyed hearing about your take on mentorship and collaboration. I think, you're right on in terms of like there is no single recipe for success, but like the influence of all the stakeholders is really crucial, and the framework of those collaborations is really key. And I think, a lot of institutes could take a lesson from Lausanne in terms of like those ideas of core funding, of a highly collaborative international team with a lot of diversity of ideas and expertise. And yeah, a beautiful scenic place to really inspire you, which I think maybe is lost in this rat race of science, is that really it is such a joy and inspiration and a playground for our imagination and creativity. So that's my takeaway from this discussion. And I really appreciate you leading me along that path. Before we're going to let you go, we got a couple of peripheral, I guess you would call them, science questions. First, if you were not a scientist, what would you be doing with your life?
Um, that is an easy thing. I would love to be involved more in music, and I could have imagined myself to be an orchestra director if I had the opportunity to study it properly. And it's was something for me that I'm very passionate about. And who knows, perhaps at later stage I might go into that direction as well.
Second life, although you never stop being a scientist, but you can add some things to the repertoire. What instruments do you play?
I play the piano mainly.
Got it. Wow. Another one of these scientists that does another thing with expertise. It was a couple episodes we talked to a guy who has a rock band going. I mean, never ceases to amaze me, the hobbies. Finally, what is the best piece of advice that you've ever been given, professionally or not? I mean, you did share that kernel from your PhD mentor in terms of leading you along the path or towards stem cells in part, but any other advice, professional or not, that's been critical to advancing your path?
Yeah, sure. There are many stuff that I heard, and they were influential in my life. And one of these things, I heard it from Tony Robbins, and he said, "Lives happen for you, not to you." And that was really important when I would have experienced the challenges in life, like all of us. And I know that it is important that I steer these challenges to my advantage rather than feeling a victim or feeling unhappy about these challenges.
Mm, that's a great one, because also it's like, look, it's all there for you. I mean, what a treat, what a gift it is, this life. And also this interview. It has been quite a pleasure, and we appreciate you sharing with us and speaking to us and our listeners. Shukry, thank you again so much. We hope to hear again from you soon. That story that should be coming out, the follow-up of your Nature Materials paper. Looking forward to seeing that in press, and looking forward to see everything that you're working on. Appreciate you, man.
Thank you very much. Thank you both for hosting me.
All right, guys, that brings us to the end of this episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and roundup papers. You can also reach out to us on X at STEMCELL Podcast or via email at info@stemcellpodcast.com with feedback or to suggest guests. We got ISSCR coming up in just a few weeks, but we have a few episodes before then, so stay tuned and we'll be back at you in a couple weeks.